This video is going to take a look at the way various molecules move across the cell membrane. So first of all, we have to remind ourselves that the cell membrane is selectively permeable, meaning some molecules can get across while others cannot. And that depends on their charge, their shape, their size. And let's take a look at how those different substances get across the membrane. So first of all, we have to look at the two major classifications of movement, passive and active transport. Passive transport does not require energy to occur. This is because it uses the concentration gradient. In passive transport, all substances move from an area of high concentration to an area of low concentration. So this includes simple diffusion, facilitated diffusion, and osmosis. Active transport, on the other hand, requires energy, and this is because substances are moving from an area of low concentration to high concentration, so they're moving against the concentration gradient. And this can include protein carriers and coupled transport. So let's start with diffusion. How does it work? Well, it's due to the random movement of particles and the fact that particles will move down their concentration gradient in order to reach dynamic equilibrium, or equal concentrations on both sides of a membrane. Once equilibrium is reached, those particles don't stop moving. That's why it's dynamic equilibrium. So as we can see here, the molecules, because of the random movement, eventually some will get through those sections of the membrane that they can fit through, causing equal concentration on both sides, or equilibrium. So an example of simple diffusion is when those particles move directly across the membrane, such as small lipid-soluble molecules, oxygen and carbon dioxide. Now those molecules can move through because they are so small. There is the odd chance that water will move through, but not enough moves through to say that water moves via simple diffusion. Facilitated diffusion. This is using a protein channel to allow polar molecules through the membrane. So normally they cannot fit through, but with a protein channel, if the molecule can fit through the channel, then it will go through. So passage is due to size, shape, charge, and this is a permanent hydrophilic channel that is embedded in the membrane. Examples can include ion channels, so allowing sodium ions through or potassium ions through. They're also what are known as voltage gated, that open and close, responding to electrical potentials in the membrane. They're also known as ligand gated, responding to chemical interactions such as hormones, so they will open and close based on the presence of another molecule. Protein carriers are another form of facilitated diffusion. Now these allow a specific molecule to bind and is then transported across the membrane. So you see a specific molecule binds, the protein then changes shape to allow that molecule through. There is still no need for energy for this to occur. Examples include the movement of amino acids, sugars, and small proteins into and out of the cell. Osmosis is the diffusion of water across a selectively permeable membrane, meaning that the water can diffuse across, but other substances or solutes cannot. So osmosis occurs from an area of high water concentration to an area of low water concentration. So if we look at this example on the left here, there is high water concentration on the left because there is less solute. There's low water concentration on the right because there is more solute. So if there's a selectively permeable membrane, keeping those purple solutes from moving back and forth, it is water that will then move to achieve equilibrium. Aquaporins. This is protein-facilitated diffusion of water across the membrane due to an osmotic gradient or difference in water concentration. So a few water molecules can fit through on their own, but not enough. So there are these embedded proteins or aquaporins that allow the polar water molecules through. So example of where you would find a type of aquaporin is aquaporin number 1 are found in the kidney, and this causes reabsorption. If they weren't there, we would urinate much more frequently than we do. So just a quick review of osmosis and solution terms, because these will come up. We've mentioned them in the past few slides. Solute is a substance that is dissolved in a solution. The solvent is a substance that does the dissolving. So for example, water and sugar, water would be the solvent, sugar would be the solute. An osmotic concentration means taking into account the concentration of all the solutes in a solution. So if we're looking at blood, the plasma would be the solvent, and the solutes would be the red blood cells, the white blood cells, the platelets. An osmotic concentration would be the concentration of all of those solutes within the plasma. So it would be 45% concentration solutes to solvent. So there are ways we can describe solutions, and keep in mind that it's all relative to what you are comparing it to. So if we're talking about a hypotonic solution, it means that the solution has a lower solute concentration. If we're talking about a hypertonic solution, it means the solution has a greater solute concentration than whatever we're comparing it to. Isotonic means that the two solutions have the same solute concentrations. So if we look at these examples here, this cell is hypertonic to the solution is hypotonic to the cell. So it's always relative what you're comparing to. Whatever has more solute is hypertonic. Whatever has less solute is hypotonic, and if they're the same, it's isotonic. So if we're talking about the inside is hypotonic relative to the outside, the cell will be shrinking because water is moving back and forth to reach equilibrium. So when we talk about those types of solutions in an isotonic medium, you're going to have equal net movement in and out of the cell of water. In a hypertonic medium, you're going to have a net movement of water out. The solute is going to draw the water out, and therefore the cell will shrink. In a hypotonic medium, it's going to be drawing the solution, or the water rather, into the cell, causing it to swell up. So now we move on to active transport. Remembering this is where energy is required, and it's usually in the form of ATP. This is because substances are moving against their concentration gradient. So movement requires specific carrier proteins because they are not naturally going to get through, and they need the energy because they're not naturally going to go against their concentration gradient. So we can have many different types of proteins, and we're going to talk about them in the next few slides. An antiporter. A carrier uses energy to move a substance across a membrane against its concentration gradient. Well, an antiporter means that things are moving opposite to each other. So there is one entity, in this case a sodium ion, that will move this direction, whereas the potassium ion will move the other direction. Noticing ATP energy is required to drive this process. So examples are the sodium-potassium pumps, and this establishes a resting membrane potential that allows our nerve to transmit impulses. So again, in this case, the sodium is going to be moving out of the cell, the potassium is moving into the cell. Antiporter, opposite directions. Coupled transporter. How does it work? Well, there are molecules that are transported against their concentration gradient by the co-transport of sodium ions or protons down their concentration gradient. So, coupled means that there are two proteins together. So we saw this one in the previous slide. This is the antiporter. But then what happens is this protein is coupled with another. So the sodium ions were pumped against their concentration gradient out of the cell, but as they return through another protein, the coupled channel, it also allows another molecule, in this case sugar, to come back into the cell. So the antiporter pumps the sodium out, they come back in passively, but allowing the sugar in. So for example, sodium and glucose uptake in the cell. Bulk transport, this means large quantities. So substances required for growth are mainly large, polar molecules that cannot cross the hydrophobic barrier created by phospholipid bilayer. So then how does our cell get those large polar molecules? Well, it's through bulk transport. The first type of bulk transport is phagocytosis. So how does it work? Particle is engulfed by the membrane. So here's the food particle that is desired by the cell. The membrane literally comes out and engulfs it. It meets together forming a vesicle, and that vesicle will travel inside the cell. So you can also see bacterium and amoebas engulf things in the same way. So the amoeba is engulfing the bacteria, phagocytosis. So an example would be ingestion of bacteria by white blood cells as well. So this is how the white blood cells fight off bacteria. Phagocytosis is like eating for the cell, taking large bites. Pinocytosis, however, is intake of fluid. It's like cell drinking. So fluid droplets, in this case, instead of solids, are engulfed by the membrane, similar mechanism as phagocytosis, forming a vesicle, and it moves inside. So this is how swelling decreases. If you ever sprain an ankle or a wrist, it swells up. Well, that swelling disappears because your cells are all undergoing pinocytosis to reabsorb that extracellular fluid. Then there's also receptor-mediated endocytosis. Endo, again, meaning inside, cytoplasm, and cis, process of. So taking things into the cytoplasm. So receptor-mediated this time, it's triggered by a specific receptor. And this forms protein-coated vesicles. So there are these receptors on the membrane. If the particular appropriate molecule binds to them, it will then bend inwards or fold inwards, creating a vesicle with these receptors on the inside. It's a coated vesicle because it's coated by the proteins. And it takes whatever the desired molecule is inside the cell. This isn't seen in cholesterol uptake. Exocytosis. Exo mean exiting the cytoplasm. So this is kind of like cell spitting. This is when vesicles from the inside fuse with the membrane and they literally eject their contents out. So you can see it happening here under a microscope. And this is how our body secretes mucus. This is how neurotransmitters are released between nerves. It is our body getting rid of larger molecules.